What's going on, Bio Kids? It's your boy, Mr. Summers. Tea time. About to enjoy some tea. I put it somewhere in front of me. COVID-19 bunker, which happens to be full of Eleanor's doctor kit and some weird blanket. Let's do this. Some things. We are going to go with a lab, like a virtual lab that's like, should be 2 days, but I'll make it 3 because I don't want you to like freak out about the amount of stuff there's to do. I asked some students in our class what if the amount of work was okay, and they seemed to think that the labs and the really long whale activity was too much in too little time. So let's put this one out for 3. If you get it done in less time than that, great. Enjoy your day. But we'll start with that. This is what we started up. It's the one that says Controlling Bacterial Lab, and it is here in this. Hey, make sure you do this attendance form every day. Takaya. All this stuff we'll get to. Look, yay, us. But Controlling Bacteria Virtual Lab is what we are doing today and Friday. Your goal on day 1 of the lab is to get your introduction done. On day 2 is to collect your data and start to like graph it out and look at it. And day 3 is to get your conclusion done. If you're a speedy person, you want to get everything done in one, your world. I'm just telling you, you've got plenty of time to do it, so don't freak out. This Bacteria Lab instructions brings up this copy of instructions 1. And if you're looking to write your introduction, the 3 slides of background information are where you'll get the information from. The lab is pretty simple, and I'm going to show you with this one. Might as well present it. Let's live a little. Ooh, fancy. All right, if you are growing bacteria on a agar plate, right? So bacteria, by now you should know, are little prokaryotic, which means single cells, no nucleus, organisms, and you can grow them pretty easily on the agar cubes you use when you do that like cell size lab where you cut them into little pieces. This one, if you grow a bacteria on that and then put on one of these discs on it, this area here has no growth of anything. And it turns out when you say discs, your iPhone 12, because you're rich, pops up like, for some reason Siri wants to talk to me about that. This area, if you look at it, stop that, Siri, you weirdo. Is the area where there's no growth. So from the disc here to there is no bacteria, and all of this whitish stuff around it is bacteria growing. So your lab is pretty simple. You're going to vary what is on these discs and see how much area they kill. The bigger this circle is, the more bacteria is killed. I'll say that again because it's probably the important part of this lab. The bigger the surface area, or this area here to here, this width, the more bacteria is killed. Okay, so this one is probably just paper, because you need some, or probably that one's probably paper. You need something showing that it is not the disc that's killing them. It's what's on the disc that is killing them. That's probably, I don't know, important. It's probably the control, and then that's pretty much it. So the lab is pretty easy. You're going to fake grow bacteria, put discs on it, and see which one kills the most bacteria, right? So those things like household cleaners, bleach solution, soap, all of them on there. We do this lab for real in actual school. It turns out, global pandemic, we got to switch it up a little bit. So these things test these antimicrobial agents that go on there, and that is kind of what you're looking for. So again, when you're writing your introduction, which should be about a little bit of background information, what are bacteria, why you're testing things to see if they actually kill them, but the whole point of the lab, which is to try to figure that out, you were looking at those things. You have, again, to figure out the independent and dependent variable. So what are you measuring? Hint, this thing right here, and then that's the dependent variable. The independent variable is probably also something to do with these discs, specifically what's on them, just in case you're wondering. The obvious controls are the same things. Time, the amount of substance you put on there, the shape of the disc. This one that has just paper. Those are the things you're looking for. And then you're going to go from there. So again, day one, if you read through that, you don't need to do the actual virtual lab. It looks like this. All you need to do is just write your introduction. If you want to stop there, go ahead. I'm going to walk you through the virtual lab. But if you want to come back to this, it'll still be here. So here is the So Fresh actual virtual lab itself. Ooh, look at us. Right? There's more information here, which is probably the same stuff that was on there. Look, objectives. Alright, so I'm just going to choose this bacteria. I'm not quite sure if it tells you to do all of them or one of them. It doesn't matter. This is Staphylococcus aureus, which is a something you get with staph infection. So that just puts it onto the plate. These little discs you put together. Disc 1, there's soap, household bleach, disinfectant, penicillin, which is an antimicrobial, like antibacteria pill you take. You've probably taken that one in your life. Amoxicillin. Again, those two are big ones to kill off bacteria. Erythromycin. Those are like the three most common like pill form of stuff you've taken in your life to kill things. And then, yay, go us. Now we've got them all marked in. Turn this mouse on, we put them in. And then the big thing is checking the zones of inhibition around the filter paper so that you can figure it out. Ooh, look, it's growing. Yay, a day is done. When you take this thing out, there will be zones of inhibition that you can measure with a ruler. I don't want to ruin the lab for you by doing it right now, so I'm not going to. But I'll look at it there and then use, yay, right? And you're measuring the area in which there is no growth. Blue is growth. No growth is the area clean. The bigger the circle, the more stuff was killed, the better the substance worked. That's what you're aiming for, right? And then to go along with it, there are sentence starters, which you can get from that information, the background information. Collect your data, which is pretty easy. Perform it 3 times. Do it with each bacteria species. So I only did that one for the staph, but make sure you do it for the Haemophilus influenza and the Streptococcus pneumoniae. That's how you say the word pneumonia, by the way. And then graph the data, like simple bar graph. All this tutorial stuff will go through, and questions in conclusion. So, day 1, you should get to here, complete the introduction to your lab report. Day 2, you should get to here, right? Do the lab, get your data, and day 3 is your conclusion and stuff like that. I'll probably hold some sort of Zoom hour in case you need it, maybe Friday or Monday, depending on how bored I get. But that's pretty much it. That's your lab. Hope you enjoy it. Bye.